Good morning, everyone. We'll probably start a talk. So it's a great pleasure to have Peter talk on the 12 Labor Seminar Series Platform 1, a framework for personalized physical modeling. As you may recall, Peter presented an overview of the 12 Labor project last November. So it's really great to have him here again. So Peter is the director of ABI. His research interests are in modeling human physiology using anatomic and biophysics-based multi-scale approach that links molecular process to tissue-level phenotypes. He is an elected fellow of the Royal Society, both London and here in New Zealand. He is executive chair of the World Council of Biomechanics and vice president of the International Union of Physiological Science. Without further ado, I let Peter start his talk. Thank you, Peter.
Thanks, Chicho. So I'm just going to start with a brief reminder of what 12 Labors is about for people that haven't been keeping track of this. So there's three platforms, technology platforms. Platform 1 is about personalized physio modeling, or a framework for that modeling. Platform 2 is about clinical workflows, getting the models into clinical workflows. And platform 3 is around the measurement side of things, put wearable, implantable, home-based sensors that we can use in conjunction with imaging technologies to help feed data into personalized models. And the three platforms, P1, 2, 3, are supporting three initial exemplar projects. The first one is on pulmonary hypertension, second on upper limb rehabilitation, and the third is on the autonomic nervous system control of organ function, particularly in relation to maternal health and digestive function. So the idea is that we use these exemplars as proof of concept for the workflows associated with those three platforms, but we're certainly planning on other projects feeding into this workflow. We're about 1.5 years into a 5-year project with this. It's a project funded by the Ministry of Business, Innovation and Employment in New Zealand. So the 12 AIs on the project are shown here. Project manager Dana and EPs are led, as you can see here, by a number of people from ABI. And in particular, for this talk, I'm going to focus on P1, and in particular, with contributions from David Nickerson, Andre, and Sarush Safai, and also from Anand, who's shown on the right here, who's part of the 12 Labors project for P1. But I also just want to acknowledge a lot of input into what we do here in terms of building infrastructure for P1 from a group that's funded by the NIH Spark project that I'll talk more about as well as I go through. But the people listed here are really providing a lot of the infrastructure that we are using for 12 Labors, building on what we've done with Spark over a number of years. So the themes of this talk are going to be, first of all, I want to talk a little bit about the fact that most diseases, particularly chronic diseases, have genetic and environmental origins, and the physics matters just as much as the genetics. And that just as evolution has exploited genetic variation via Darwinian natural selection, so through the 500 million years since the Cambrian explosion of development of human physiology, physiology has exploited physics. And I want to show you how that's pushed to the limit, in fact, of what physics can allow. So the body is a highly integrated system in both a multiscale and a spatially connected sense. Our models address the multiscale gene, protein, cell, tissue, organs challenge, but we've not done a lot in terms of looking at the interaction between physiological systems. So how do we create a modeling infrastructure that can integrate these multiscale models into whole body physiological systems? I think we need new algorithms that assemble semantically annotated models. And what I'm going to talk about is more posing questions and answers at this stage, but I just want to use this opportunity to get us thinking about what we need to do in order to really deal with whole body physiology rather than physiology that's just geared to one particular organ or even organ system. So I'm going to start by thinking a little bit about genetics and talking a little bit about genetics and genome-wide association studies, or so-called GWAS. So the human genome, which has been around for about 20 years in terms of the initial sequence, but only in the last year or two has the sequence been completed. The human genome has 3.3 billion base pairs, of which 1.2% only provides the 23,000 protein encoding genes, which are organized into the 23 pairs of chromosomes. So the other 98.8% includes, it's not only, but it includes the regulation of transcription. So this is a surprise. I think when the human genome was first published in early 2000s, I should say, it was seen as the blueprint for life, and that we'd be soon discovering a one-to-one mapping from genes to particular dysfunction or phenotype, physiological phenotypes, and therefore the dysfunction of those phenotypes associated with disease. That view has totally changed, and I want to just provide some of the evidence for that. So one of the sources now of our understanding of the relationship between genomes and disease are these so-called genome-wide association studies, GWAS, where you take a large number of individuals and you sequence the whole genome, and you look at a particular condition, and you look at individuals who have that particular condition, and you compare them with normals, and you look for the differences. Because that gives you the heritability of the genes associated with predisposition towards a particular disease. And I'm using rheumatoid arthritis as just an example here, but it could be any disease. So the protein coding, remember 1.2% provides the genetic inheritance associated with proteins that are coded for by the genome for this particular disease. But it's the rest of the genome, the non-coding regulatory part, that in fact is found to be just as important or more important in many cases. And I'll give some examples. So we really are reaching an alternative view that we had in the early 2000s, that it was one gene, one function, one phenotype, to now the point where every phenotype depends on every gene. So the whole genome becomes basically involved, and all the evidence from many studies is that when you look at the impact of a given bit of DNA on a particular condition, it's minuscule. It's the whole system it's the sum of a large number of small effects from a very large number of parts of the genome. So one interesting observation, and that's this paper shown below by Boyle et al. From 2017 in Cell, is that the heritability contributed by each chromosome, in fact, tends to be proportional to the physical length of that chromosome, which is a pretty good indication that it's simply the amount of DNA it's not specific genes, it's just the total. And their conclusion is that complex traits are mainly driven by non-coding variants that presumably affect gene regulation. And one example of this, it's a sort of extreme example really, but there's 100,000 single-letter DNA variants that influence a person's height, each one with a minuscule impact. So if we think about the relationship between the dependence of phenotype on genetics versus physics, and by physics here, I'm really thinking about the environment that influences physiology, and the physics is quantifying that, underpinning and quantifying that environment. So if you think about a vertical scale where you're going from monogenetic to omnigenetic, where omni means the whole genome and mono means just one gene, and then on the horizontal scale going from zero physics to 100% dependence on physics and the environment, you could say that Mendel's P's, that's the classic experiments that showed the heritability of a particular gene on the function, the phenotype, namely the type of P, that would be sitting at the bottom left here. It's close to monogenetic and close to very little influence of the environment. I mean, it's got to be nurtured, of course, you've got to have food supply, but it's an example of a relatively very low dependence on both physics and typically one gene. Height, on the other hand, the example I just gave, it depends just about on the whole genome. I mean, there's obviously some impact of physics because, you know, all the nutrition that you have does affect your height, but it's a relatively small effect. But the genetic inheritance associated with height is, in fact, very widespread through the genome. And then if you look at the various diseases, and this is the data coming from that particular paper, but there are many papers now, and it's widely accepted that most diseases depend on most of the genome. And I won't go into the details here, but these particular ones have been quantified in relation to the very careful studies with very large numbers of people in these gene-wide association studies. And the analysis of that data is showing the very broad dependence across the genome, as well as some unique dependence on particular genes, protein encoding genes that are involved in particular pathways for these diseases. So that's the background. We've got this combination of genetic inheritance and an enormous dependence on the physiology, which is associated with the physics of how the whole system works together in a highly integrated sense. And the other thing that's happening a lot at the moment is the NIH, which is a $36 billion public health funding agency, the world's largest funding agency. They've set aside about $1 billion a year for what they call the Common Fund, which is under the direction of the director and is not handled. The rest of the money is handled by individual institutes, but the Common Fund is under the direction of the director's office. And recently they've set up something called the Common Fund Data Ecosystem, CFDE, which we're part of. And that's really beginning now to provide a platform for assembling data from multiple aspects of measurements on humans. And one of them is Spark, which we've been part of for about 5 years. So this is the autonomic mapping, the autonomic system, and the ABI is involved here in developing the scaffolds for mapping spatial data coming from experimental groups that are measuring all sorts of aspects of autonomic function. And if you visit the website, you'll see our scaffolds from ABI being used to underpin spatial mapping of data within organs, but also across organs. And I'll talk more about that later. Then another project is HubMap, which is about the mapping of human cell types right across the body. We've got a subcontract in this project as well, and we're looking at the mapping of HubMap cell data through our spatial scaffolds to the Spark scaffolds, spatial scaffolds for handling information about tissues within organs. So HubMap has this goal of mapping healthy cells in the human body in a spatial sense. So again, being able to provide huge amounts of data that will help in understanding the complexity of the integrated function of the physiology of the body. Another one is metabolomics. This is led by Shankar Subramaniam, who's a colleague of ours at UCSD, and this will be, I think, another important part of the picture, again, funded through the Common Fund. And this one is actually separate, but it's the Human Cell Atlas Project, which is a very large community now from many countries who are beginning to assemble data about human cells and the spatial distribution characteristics and so on. So we need to be plugging into that. And the MB 12 Labours proposal is funded under the Catalyst Fund of MB, which means that it's got a very strong international focus. And we're expected as part of this project to be engaging with other international projects, because I mean, what we're tackling here is something that requires a global effort, and we really need to be consciously linking into many of these large-scale efforts that are happening around the world, with in many cases much larger funding sources than we have available, but we'll be able to leverage data and models and information coming from those other projects. And I thought I'd just mention one particular recent publication a couple of weeks ago in Science, which is looking at single cell transcriptomics. And now there's phenomenal ability to pull out the understanding of the genetics of individual cells taken from human tissue. It's called the Tabula Sapiens Consortium, and they've taken 500,000 cells from 24 different human tissues and organs, and they're characterizing 400 cell types, which is a very good range of cell types in the human body. And again, just data that we'll be able to plug into our frameworks. Okay, and now I want to just have a couple of slides that really illustrate why I think physics really matters. I mean, I'm speaking to the converted, I think, in terms of ABI, but in terms of worldwide physiology or the bioscience community, there's really a lack of understanding of the importance of physics. It really does matter just as much as genetics. And I just want to illustrate a couple of particular examples of that. So in your body, you have about 10 to the 15 mitochondria. They're producing the ATP that powers your body. And the energy flows in the body are truly astounding. And the mitochondria, of course, are a bacterial, started life as a bacteria that got incorporated into human cells at a very early stage in evolution in the 500 million year evolution of humans from the Cambrian explosion. And if you look at the amount of area, the key structure in mitochondria is the inner cell, the inner mitochondrial membrane. And if you took those 10 to 15 cells and you multiplied that by, for one individual, remember, multiplied that by the area for each mitochondria, you get about 14,000 square meters. So that's the size of 4 football fields, right? So in your body, in order to produce the energy you need to drive your daily activities, you've got a surface area of 4 football fields that are covered with proteins that are involved in transporting protons across the inner membrane of the mitochondria. And that proton gradient is used to drive the ATP synthase process, the F0, F1 ATP synthase. And that's then generating the ATPs that become the energy currency for all cells in your body. And that process is driven by the oxygen electron transport chain, driven by oxygen and involving this transport of protons across these cell membranes. And that, if you take that full mitochondria, that 14,000 square meters times the density of these pumps, you pump protons across that membrane at 10 to the 21 protons per second, which is just almost unbelievable. That's the number of visible stars in the visible universe per second across the inner membranes of your mitochondria every second. Those little ATP synthase are little generators spinning at about 6,000 RPM. So every cycle they go through, they generate in 100 milliseconds, they generate 3 ATP. And those energy flows are essentially driving your ability to sustain life. And if you look at the gradient, the energy gradient across that inner cell membrane, it's about 180 millivolts across that inner mitochondrial cell membrane. That membrane is 6 nanometers thick. So if you divide 180 millivolts by 6 nanometers, you get the energy gradient that's needed to drive those ATP synthase to generate the ATP that we need. That gradient, 180 millivolts divided by 6 nanometers, gives you 30 million volts per meter. That's about the energy in a lightning strike. So again, this is not genetics, this is the physics associated with being able to provide you with the energy you need to drive life. Now let's look at how physics is pushed to the limit. As people that work with me know, I'm a little bit passionate about units, and I think what's really interesting is that the best units to use for physiology, there's 3 of them. You only need 7 units to define everything that we do. Those units consist of joule, seconds, and meters, which define the space, time, and energy environment in which we live. Then you have 4 units, coulombs, candela, moles, and entropy, which essentially set up the link with microscopic processes and give you the macroscale units that we need for defining the physics of these processes. And there's a bunch of physical constants associated with that link from microscale, Avogadro's number, for example, up to macroscale. So if you start with coulombs, essentially coulombs is counting electrons through Avogadro's number. You're counting the amount of charge on an electron and scaling it up via Avogadro's number. And the body can detect 1 electron. It can detect the movement of 1 charged ion through a molecular membrane ion channel. If you look at photons, our eyes can detect, well, some eyes, but ours in particular, can detect 1 photon. That's the limit. There's no point in going beyond that. Evolution has gone as far as it's possible to go in terms of the limitation of the physics. If you look at our ability to smell, and a good example here are sniffer dogs who have around 300 million olfactory sensors and about 50 times the sense capability of humans. They can detect 1 part in 10 to the 12, so in concentration. So they're smelling individual molecules, 1 atom. Again, you can't go further. And I haven't got an example of this, but it'll be the same with entropy and probability states. So evolution is pushing genetics through Darwinian evolution, the evolution of function related to genes, but equally it's pushed physics to the limit that's possible in the laws of physics as we understand them. So now I just want to have 1 or 2 slides on before I get into how we deal with across different organ systems, just talking about the ABI multiscale modeling framework, which is developed over a long period, really, over 20 years. And it's all about reproducibility and modularity. And as many people will know, we've got standards now called SALAMAL around how to encode models at these different scales, and in particular this example showing the incorporation, for example, of sodium channels. So individual proteins with their gating processes can be imported. Each one can be a model in its own right, can be imported into a channel, and those channels combine to give you a model, for example, of a Hodgkin-Huxley ion channel. Or another example, and this is one coded up by Nima in ABI on the glucose uptake in enterocytes, where in this case, the individual SALAMAL models represent all the processes associated with transport of glucose across the membranes of these epithelial cells. And then they get imported into, with appropriate annotation, get imported into higher level models that can deal with glucose uptake in these epithelial cells, and that the entire cell is shown on the left for these transporters. And at the bottom is the link to the Frontiers Physiology paper where this was described, but also the Physiome paper where there's a complementary Physiome paper that describes the detailed models associated with the physiology of this process. So here's an overview of the multiscale modeling framework that is based on these standards, from SALAMAL through to organ systems, and the whole body. But I just want to, the reason I put this slide up is I just want to mention this concept of the FTU, the functional tissue unit, because it's a concept that Bernard de Bono, who works with us, formed a number of years ago, and he and I wrote over nearly 10 years ago now on how functional tissue units, FTUs as we call them, are a crucial part of thinking about how molecular biology and cell biology emerges into physiology. So physiology depends on genes and proteins and particular cell types, but the 3D organization of those cell types, and very often it's a small number of cell types, but their 3D organization within a small piece of tissue is what really defines the phenotype for that tissue. So FTUs are going to be a very important part of how we think about organs and organ systems in this whole body approach. And the same process of assembling proteins into cellular models into FTUs, FTUs into organs, all of this is going to depend on that same infrastructure that's been pioneered by David Nickerson and others, Andre, to make sure that we've got a properly semantically based ability to assemble models into higher level structures, which is going to be absolutely key to the development of the whole body models that we'll talk about. So one example of the FTU, just giving one example from the cardiac work led by Martin Nash at ABI on modeling various aspects of mechanics and electrophysiology. And of course, many people in ABI involved in different functions of the heart, and all the work on tissue function led by Bruce Smale and others on being able to bring these into these whole cell models, and then into these whole organ models, and then the dependence on the cellular processes shown as the third example here. What we need to get better at doing, and we have started, and I think in particular of the work of Gonzalo and Sarush with Marty in terms of being able to capture those high-level organ finite element-based spatially distributed models that then get captured through AI, machine learning techniques into surrogate models. And the end result of that is a black box that has inputs and outputs that you define in order to represent the function of that organ in place of that very complex, expensive, detailed finite element model, but always with the ability to go back to your detailed finite element model. But if we're talking about the use of these models in clinical practice and large-scale incorporation into organ systems and across the body, we've got to be thinking about how we deal with these models in the context of model reduction and black boxes that through machine learning algorithms become a way of capturing that function through to the larger systems. So if you think about that black box for the heart in this case, it's got certain inputs like venous pressure through the vena cava, pulmonary arterial pressure, pulmonary venous pressure through to aortic artery pressure. I mean, it is a pump. It's all about pumping blood, and flow and pressure are the key elements. And of course, it's got to be supplied with its own energy supplies. So the coronary arterial blood flow in and out are additional inputs and outputs, which are bringing in nutrients and hormonal control of the cardiac muscle. But also it's got all these nerves that are involved in regulating that function through the autonomic system. And there's an interesting difference here, because when you talk about a blood vessel into the organ and blood vessels out, it's a very small number. You know, it's only a few vessels that represent all the inputs and all the outputs for blood flow into and out of the heart. But when you think about autonomic control, every neuron which is monitoring the behavior of some part of the myocardium or is controlling some aspect of the myocardium, you know, at the level of the sinoatrial node or the AV node or some part of the tissue, is dealing with an individual neuron, a sympathetic or a parasympathetic or a sensory neuron. So you have a huge number of inputs and outputs associated with those neurons, unlike the vasculature. in many ways, the most complex inputs and outputs for the heart are the neural inputs and outputs. So as we build our functional tissue units within these organs, and here I've just indicated FTUs for the right atrium, right ventricle, left atrium, left ventricle, the right atrial ganglionic plexus, which is where a lot of the integration is happening in terms of neural inputs, and then a vascular bed where you're dealing with the internal distribution of blood within the heart. So we've got to understand how these functional tissue units within the organ operate to achieve the black box description of the whole organ with its well-defined, semantically defined inputs and outputs. And I mean, I'm using the heart as an example, but this obviously applies to every organ and every aspect of physiological function across the body. So we have a standards-based framework, cell-ML, field-ML. We have energy-based mass framework, bond graphs, Port Hamiltonians. We know how to deal with spatial aspects. The work that Richard Christie has done in building a framework for scaffolds is hugely important, I think, globally, in terms of the way that through our connections with NIH, we're going to be able to handle a lot of the spatial distribution of data that comes back through all the NIH projects. And there is no other group in the world that can handle the spatial aspects in the way that we can. And I think it's a tremendous tribute to the group in ABI who've really facilitated this framework. But we need, and what I want to talk about now is we need much better ways of handling the complexity associated with the networks that relate to systems physiology. So I'm now going to switch from the multiscale modeling frameworks that we've dealt with in the past to thinking much more about where we're going in the future with systems physiology that's crucial to 12 labors. So how are the systems of the body connected? And I have to say, it's a question I've only asked myself fairly recently, which is a bit embarrassing. But so the first point is that every cell has nearly the same genome. I mean, obviously with any given cell is typically the product of only about 5 cell divisions, somewhere between 3 and 7, but typically around 5 cell divisions to form that particular cell. And those cell divisions do have mutations. So there are small differences in the genome between cells, but it's almost the case that every cell has the same genome. So that itself is the beginnings of a highly connected system on genetics. Then let's look at the gut. In many ways, the gut is the most interesting. It's the first organ. I mean, that's where we all started from 530 million years ago when we began life as a worm, essentially, with a gut. And food input at one end and output the other. And that's what began the evolution we can trace our ancestry back to. And that gut, as it turns out, has its own enteric nervous system. So the gut is really the beginnings of human beings. And that gut had to be protected. So it's got from the outside world. So it ends up developing an epithelial integumentary system, the skin. And I'll talk more about that in a minute. And then you've got to supply nutrients to that. So the cardiovascular system developed, also controlling heat flow. And again, these are just the systems that are thinking about what is it that couples the whole body together. So cardiovascular, clearly, and along with that, the lymphatic system. So the lymphatic system is dealing with the larger molecules that don't go into the bloodstream. So all the fats from your digestive system get passed into the bloodstream after they've gone via the lymphatic system. So it's a highly interconnected system in the body, but closely linked with the cardiovascular system. So it definitely is one of these whole body connections. And then, as I've mentioned, the autonomic nervous system is crucial for regulating every visceral organ is innervated with nerves that are undergoing parasympathetic or sympathetic control and constant monitoring, talking back to the brainstem and talking to each other via the brainstem and via the enteric nervous system of the gut. And then the musculoskeletal system, of course, gives us locomotion. And force transmission becomes another way of connecting the whole body. I mean, all your muscles and bones that transmit loads from the feet up through the body, including gravity, of course, are meaning that you're coupling every cell is mechanically sensitive. So the musculoskeletal system is another example of a highly connected system for the whole body. And I thought I'd just mention here, reminding us about the three motor systems that control the function of physiology. So on the left is the somatic system, which is individual neurons forming synapses with the muscle, going from the central nervous system, from the motor cortex, out to individual muscles. The autonomic system, with its two branches of sympathetic and parasympathetic, and also its enteric system, of course. But the autonomic talking to the brainstem through two neurons, pre-ganglionic on the sympathetic side, going via sympathetic ganglions, which often sit either adjacent to the spinal cord or somewhere close but outside the organs. Typically, the two neurons involved in the parasympathetic, the synaptic junction between pre and post is occurring in the organ itself, not always, but mostly in the organ itself. But again, only two neurons, and then that's a paracrine signaling system. And then finally, the neuroendocrine. So from the hypothalamus and the brain controlling the pituitary gland, releasing hormones into the bloodstream, is then becoming another global way of achieving motor control right around the body. Okay, let's start now with these organ systems that are involved in whole body control. Digestive system. So, we have a GI group who are developing in the ABI and part of 12 Labours who are developing models of many parts of the system. And at some point soon, we've got to think of this as one system, the whole connected GI system with its autonomic control and sympathetic, parasympathetic, and also its vagal parasympathetic inputs. So we now have all the connectivity that we need to be able to understand the autonomic control of that whole body system. And as part of the SPARK project, we've also been mapping the neurons in individual organs, in this case, the stomach, that then are coupling. And the data is coming through now too on the coupling between the connectivity from the brainstem to the organ and how that couples to the neural systems within the particular organ, in this case, the stomach, but of course, across all the organs of the GI system. And then if you look at the functional tissue units, we need a functional tissue unit that represents the tube, what's going on in a tube that will be parameterized differently for different parts of the GI system, but is essentially in its structure an FTU that exists everywhere in the GI system. And that tube, that simple cylindrical tube with its ability to contract, smooth muscle contraction, it's got a number of networks of muscle fibers for longitudinal, circular, and a network of intrinsic sensory neurons. And then in cross-section, it's got this fascinating nervous system connecting top to bottom, connecting the gut in a way that's global, whole body, and yet stands completely separate from the autonomic system, highly interconnected with it, but nevertheless a complete tube of neurons in the submucosal plexus and in the myenteric plexus. And then another FTU that sits at the level of exchange processes and the connection with epithelial cells. And I think essentially this is an exemplar for going from a whole organ system to the FTUs that you need at the level of one level of function, but need to include FTUs that exist in detail and connect you with cells at the level of exchange processes within the FTU, just for this particular organ. The autonomic system is just a figure here showing the level of connectivity into all parts of the visceral organs of the body from the brainstem. And the interesting thing about the autonomic system is it's hardly changed in 200 million years. If you look at the autonomic system as we are doing for the Spark project across, in particular in the Spark project, we're dealing with the mouse, the rat, the cat, the pig, the human, I missed any. And there's very little difference, very subtle differences, but essentially the same autonomic system across all species. And shown here are some of the autonomic nerves innervating the heart scaffolds. So there's the data here coming from groups that we're working with in Spark, but then incorporating those data into the scaffold and illustrating the extraordinary neural innovation that exists in the ventricles of the heart. So you can't ignore the neural inputs into cardiac function. They're clearly hugely important, both in sensing and controlling aspects of cardiac function. So, and if you wanted a reminder about the physiological mechanisms that are controlled by the autonomic system, so all of this is happening outside your voluntary control, there's a list, and that's not even starting to look at pathological events like vomiting, for example. That's just saying this is all the stuff going on all the time in your body as an integrated system facilitated by the autonomic system. So we really have to start taking it much more seriously, I think, than we have in the past. Okay, so the second organs that we've talked about, the gut and the ANS. The third one is the skin. And the fascinating thing about the skin is that we think of the skin and the epithelial layer, or the epithelial cells associated with the skin, as this external layer to the outside world. And epithelial cells, of course, have evolved to provide that barrier. But the skin is only a couple of square meters in size. That's the size of a table tennis table. So keep that in mind, that your skin is about the area of a table tennis table. Now start thinking about the way that the skin epithelial layer penetrates inside the body. The lungs, Marin's favorite organ. Here's the area of the epithelial tissue that again is providing a barrier to the outside world, but now a barrier to all the particles that you're ingesting when you breathe. So it's happening inside you, but is about 80 square meters. So it's about the size of a squash court. So a lot bigger than the skin. Now the gut, top to bottom, all that surface area is about the epithelial tissue again, protecting you from the environment, is about 200 square meters, and that's about the size of a tennis court. Finally, the kidney, where all your exchange processes go on between the outside world, what you need to take out of your body, and the endothelial, the transport of your circulation. The kidney, the area is 400 square meters. So that's about the size of a basketball court. So when you think about epithelium and that protection, don't just think of 2 square meters of skin. Think about, 400 square meters of kidney protecting you again from the outside world, or 200 square meters of gut, or 80 square meters of airway epithelium. Right, so now how do we begin connecting all this together? So I decided that we needed a new term here, which I'm calling functional connectivity. So FC maps, FC for functional connectivity maps. So we've got, and here I'm just going to give you the broad regions that deal with the organ system. So we've got the digestive system. We've talked about the integuum, the skin, supporting the epithelium, with all its penetration inside the body as well. Is the second system. Third one, in terms of evolutionary, so I'm listing these in the order of what I'm presuming is evolutionary development, although an evolutionary biologist might want to correct me on the order here. But anyway, third is probably sense organs. As you moved around as a worm with a gut and some sort of external protection, you needed to sense the world around you with sense organs, and that was the beginning of the brain. brain and nervous system. So you need a lot of area here for dealing with brain function, and probably a lot more than I've allowed for so far. And then also, as part of the nervous system, on top of the digestive system, I've got these regions of enteric nervous system connectivity in the gut and for both my enteric plexus and the submucosal plexus. So they become part of the neural connectivity, but now standing alone at the moment in their enteric nervous system. And as you'll see shortly, highly connected into the autonomic system. Next is cardiovascular cardiorespiratory. I mean, the cardiorespiratory system is all about oxygen transfer from the air to tissue. So it's ridiculous in a way to be thinking about the heart without also thinking about the lungs and the circulation, because they're all involved in getting oxygen from air into your cells. And that's why there's an additional piece of the epithelial cells. Here's the lung tissue sitting up here for the airways, epithelial cells of the airways. Then the musculoskeletal system, and I've actually had to divide this in two. I need to talk more to Tor about this, but and I'll show you why in a minute. But really one system in terms of locomotion, but there's a lot more muscles than are just locomotion. So the muscular system has a number of muscles. I mean, breathing is one example. So we need to be thinking about muscular system from a point of view of not just locomotion. Urinary system, which we've talked about, reproductive, male and female, endocrine and immune. So those are the 12 organ systems that give rise to the name 12 labours for our project. And we're not dealing with all of them. We're going to put immune, probably endocrine, on a lower priority for a while, and we're dealing with some of the top ones first. But in the end, we've got to be able to couple all of these systems together. So what I'm thinking about with these functional connectivity maps is, how do we do that? How do we provide an environment where we can really be thinking and developing the semantics that allow us to properly deal with assembly of models for the whole body, from genes through proteins and upwards for the whole body. So what we've been doing is creating this functional connectivity map and dealing with, initially, there's a lot of work still to do, but we've got most of the autonomic system in here now, and I'll show you in a minute, zooming in, but this is being done with a technology that allows us to, and this is very much the development of Dave Brooks, in terms of using street maps, Google Street Maps technology, for being able to deal with zooming in and panning from the big picture right down to whatever level of detail we need to go down to, and I'll show you that in a second. But what we need to do in this big picture is to include all of the functional tissue units right across the body that we need to be dealing with in our multi-scale modeling framework, but have them all well-defined with inputs and outputs at this level of the whole big picture of all the organ systems. And that means that we can put the whole of the cardiovascular connectivity, for example, into the map under the cardiovascular system, organ system. So here, for example, just showing in more detail for some of the muscles in the arm, but this is being done for all muscles in the body, just providing the vascular connectivity of each of these muscles, where the idea is that every named blood vessel is labeled on this FC map, so that once you've got through your defined semantically labeled blood vessel down to that point, then you're dealing with an internal network of blood vessels that sits inside that organ or inside that particular muscle in this case. Yeah, well, it is an organ inside the muscle, which is sitting inside a larger unit for function of the locomotion. But from an organ point of view, all of that vasculature is within that organ, and that's fed by typically one artery going into that particular organ. So the functional connectivity map, has to identify every vascular connection, both arterial and venous, of course, and likewise the lymphatics and the neural connection into all of those individual muscles. And then from that, we have the ability to pull out, automatically process that semantically labeled map into a form that can be used to form the bond graph frameworks that Sarush has been developing for looking at whole body cardiovascular blood flow. And a little example of a bond graph model shown there, but from which you can then do using the bond graph framework and solving the whole body system, being able to then give you pressures and flows for an individual based on identifying the appropriate parameters for the cardiovascular system for an individual of a particular body shape, etc. So this is an example where we're going from the connectivity defined by this functional connectivity map all the way through to a functional output in the form of the pressures and flows in the circulation system. And then just going into so this is now with all those neural connections for the autonomic, sympathetic, parasympathetic, and sensory function. So Dave Brooks processes this through to a database that then automatically creates a environment where we can zoom in to any level of detail and look at, for example, all the connectivity that's existing within the spinal cord or the parasympathetic or the prevertebral ganglia or the paravertebral ganglia, such as the stellate, and then have all the sensory information coming back from the sensory nerves that are then, so all of these are now semantically labeled, and all of those labels are pulled out automatically from that previous FC diagram that I showed. So the FC map becomes an environment that can contain the whole of the big picture and can contain all the semantics, allowing you to automatically create the connectivity and the functional tissue units that we need to get into our multiscale modeling framework. And that will give us the ability to define the models that we need at the level of the brain stem. The NTS and the nucleus, various nuclei, nucleus tractus solaris, which is the one getting all that input from the sensory system, nucleus ambiguous giving you the parasympathetic outputs and so on. And I'll talk more about that in a second, but this is where we've got the ability to set up all the mapping between the sensory side and the motor control side for the brainstem that's going to be vital as part of this whole body autonomic control. And of course, these are going to talk upwards to the brain, and there'll be lots of function going on at the brain level, including the hypothalamus and pituitary for the endocrine system, where those models of the connectivity can be detailed on this type of map. And those maps, by the way, can also provide model interfaces. We can use them to set up the models for specific physiological questions that we're asking. And they provide the way of then semantically generating the data, for example, in the SPARK project where we're dealing with multiple species, and also generate the connectivity to the 3D body, because this is another part of SPARK, is the whole body system. And our challenge here is looking at homeostasis, the ability to really understand physiology for controlling homeostatic mechanisms in the body, defining the connectivity, defining the reflex loops, creating these multiscale models, and then beginning to really deal with the control systems. And Finbar has been, in the ABI, has been developing what are called Koopman operators for projecting nonlinear control up to a larger linear state space, for being able to use linear control theory for helping us understand control in these systems. And then thinking about disease in terms of perturbations of parameters of these models in the physiological state space. I'm going to do this pretty quickly because I'm running out of time, but a great example of this is the carotid body, which sits at the top of the common carotid artery, is highly vascularized and has all the neural inputs for sensing all the various components in the bloodstream that are needed as part of these control loops. And there's a really beautiful paper by Julian Patton and colleagues, which looks at the mapping from the cells in the carotid body through the cell bodies and the petrosal ganglion through to the brain stem, and then how each of them forms these reflex loops out to controlling particular aspects of physiological function. So the complexity is not nearly as bad as it would seem because of the fact that these reflex loops are dealing with control of very specific quantities for homeostasis. So we've got to understand what those loops are and how to model the control processes between the sensory inputs and the motor outputs. And I think I'm going to skip this, but there's a really interesting way of looking at the connection between the cell types and the nerves and the individual neurons within the fascicles, but I think we've run out of time. So in conclusion, we need to model systems physiology, but based on standards and modularity. We need reproducibility. We still need all the frameworks that we've developed for reproducibility. And I think COVID has demonstrated a very nice example of this need, in that you have a genetically identical virus, say the Omicron virus, which has got, turns out, enormous variability across the population in terms of susceptibility, in terms of the symptoms that are exhibited. And we need to understand the subgroups, the physiological subgroups and the individual susceptibility that comes from the combining genetics with physics for understanding people's susceptibility to a single genetic invasion in the form of that virus. So thank you.
Thank you, Peter. It's how I put it. It is really it is a comprehensive, high-level, informative, insightful talk. Really, the best talk every time.